वेलकम टू ऑल इन रेडियो खांची 90.4 FM, हम सबका रेडियो. I, Dr. Rupa Verma, faculty from MSc Biotechnology, Department of Botany, Ranchi University, Ranchi. In my previous lecture, I had discussed about the biomolecule carbohydrate and its classification. Today, I will discuss about the general process of metabolism and the process of glycolysis. How 6 carbon containing molecule that is glucose involves in central position in the metabolism of plants, animals and many microorganisms. This lecture will be useful for all UG and PG students of biological sciences like biotechnology, botany and zoology. Books I refer for this topic are Biochemistry Leninger and Biochemistry Strayer. Now start with metabolism. Metabolism is a series of interconnected chemical reactions occurring within a cell, and the chemical compound involved in this process are known as metabolites. Metabolic pathways fall into two categories. First one is anabolic pathways. Anabolic pathways are involved in the synthesis of compounds and endergonic in nature. The second one is catabolic pathway. Catabolic pathways are involved in the oxidative breakdown and are usually exergonic in nature. Some pathway can be either anabolic or catabolic depending on the energy condition in the cells. They are referred to as amphibolic pathways. In this lecture, I will describe individual reactions of glycolysis and also describe the various metabolic fates of the pyruvate produced by glycolysis. Now come to the term glycolysis. Glycolysis is an oxidative process and is also known as Eden-Meyerhof pathway. Here glyco means glucose and lysis means breakdown. This pathway occurs in the cytosol and is active in all tissues and does not require oxygen. This is important for ATP production. During the sequential reactions of glycolysis, some of free energy released from the glucose is conserved in the form of ATP and NADH. Glucose is relatively rich in potential energy and thus a good fuel. It is not only an excellent fuel, it is also remarkably versatile precursor capable of supplying a metabolic intermediates for biosynthetic reactions. In animal and vascular plants, glucose has four major fates. First one, it may be used in the synthesis of complex polysaccharides. Second one, it may oxidize to 3 carbon compound through glycolysis to provide ATP and metabolic intermediate. It can be oxidized through pentose phosphate pathway to yield ribose 5-phosphate and NADPH. Glucose through this process, pentose phosphate pathway, NADPH produced, which is used in reductive biosynthetic processes. So, glycolysis is an almost universal central pathway of glucose catabolism. The glycolytic breakdown of glucose is the sole source of metabolic energy in some mammalian tissues and cell types, as for example, erythrocytes, brain and sperm cells. Some plant tissues that are modified to store starch and many microorganisms are entirely dependent on glycolysis. Fermentation is a general term for the anaerobic degradation of glucose or other organic nutrients to obtain energy. Because living organisms first arose in an atmosphere without oxygen, anaerobic digestion or breakdown of glucose is probably the most ancient biological mechanism for obtaining energy from organic fuel molecules. Before describing glycolytic pathway, we should know about the structure and bond energy of ATP. So, come to the ATP description. Organisms are constantly making and breaking ATP. A complete description of these chemical processes is metabolism, which has essentially two parts, catabolism and anabolism. Catabolism leads to release of chemical bond energy which goes in part to synthesize ATP. Anabolism utilizes the energy trapped in ATP to synthesize new molecule. The standard free energy of hydrolysis of ATP at pH 7 and 25 degrees centigrade is minus 7.3 kilocalorie per mole. In most reactions, energy donated by ATP involves group transfer of phosphate group or AMP but not hydrolysis of ATP. Simple hydrolysis of ATP will only result in the generation of heat energy and will not be useful in driving a chemical process. This means that the part of an ATP molecule transiently forms a covalent intermediate in order to contribute its free energy to derive a chemical process. Now I will begin with the description of glycolytic pathway. This pathway is common to all cells, both prokaryotic and eukaryotic. In eukaryotic cells, glycolysis takes place in the cytoplasm. So, the breakdown of the 6-carbon glucose into 2 molecule of the 3-carbon pyruvate occurs in 10 steps. The first 5 steps of which constitute the preparatory phase. The first phase converts glucose to 2 molecule of glyceraldehyde 3-phosphate. The second phase produces 2 pyruvates. So first phase involves cleavage of 1 hexose to 2 triose. Second phase of glycolysis involves generation of 2 ATPs, 2 NADH and 2 pyruvates. In the first step of preparatory phase, glucose is phosphorylated at the hydroxyl group on C6. The glucose 6-phosphate thus formed is converted to fructose 6-phosphate. The enzyme hexose kinase is involved when glucose is converted into glucose 6-phosphate. Here delta G is large and negative. This is a priming reaction where ATP is consumed here in order to get more later. ATP makes the phosphorylation of glucose spontaneously. Two major forms of enzymes work here. These are hexokinase and glucokinase. Hexokinase is found in all cells, but glucokinase found in liver cells. Glucokinase functions when glucose levels are high to sequester glucose in the liver. But hexokinase is regulated allosterically. The enzyme phosphoglucoisomerase converts glucose 6-phosphate to fructose 6-phosphate. In third step, phosphofructokinase converts fructose 6-phosphate to fructose 1,6-bis-phosphate. This is the second priming reactions of glycolysis. This is committed step and large delta G. That means PFK is highly regulated. In fourth step, the enzyme aldolase converts fructose 1,6-bis-phosphate to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Here, by this enzyme aldolase, hexose cleaved to form 2 trioses. This is the main lysis step that gives the pathway its name. The dihydroxyacetone phosphate is isomerized to a second molecule of glyceraldehyde 3-phosphate in step 5 by using the enzyme triose phosphate isomerase, that is TPI. Conversion of DAP, that is dihydroxyacetone phosphate, to glyceraldehyde 3-phosphate by TPI maintains steady state. Here ends the first phase of glycolysis. To summarize, in the preparatory phase of the glycolysis, the energy of ATP is invested. So, in this stage, we can summarize the type of reactions in 5 steps. Step 1 is as phosphorylation reaction. Step 2 is isomerization reaction. Step 3 is again phosphorylation reaction. Step 4 is cleavage reactions. Step 5 is again isomerization reactions. The important is that for both phosphorylations, ATP is the phosphoryl group donor. All the sugar derivatives in glycolysis are the D isomers. The energy gain comes in the payoff phase. This is the second phase of glycolysis. In the second phase, metabolic energy produces 4 ATP. Net ATP yield for glycolysis is 2 ATP. In the sixth step of the reaction, glyceraldehyde 3-phosphate dehydrogenase converts glyceraldehyde 3-phosphate to 1,3-bis-phosphoglycerate. Inorganic phosphate is used as phosphate donor. NADH generated in this reaction is reoxidized by respiratory electron transport chain. The seventh step of this reaction involves the enzyme phosphoglycerate kinase, that is PGK. This type of phosphorylation here we may refer to as substrate level phosphorylation. The enzyme phosphoglycerate kinase converts 1,3-bis-phosphoglycerate to 3-phosphoglycerate. In the eighth step of the reaction, where phosphoglycerate mutase converts 3-phosphoglycerate to 2-phosphoglycerate. Here, phosphoryl group moves from C3 position to C2 position. Mutases are isomerases that transfer phosphate from one hydroxyl to another. In the ninth step, the enzyme enolase converts 2-phosphoglycerate to phosphoenol pyruvate. This is near equilibrium reactions. Energy content of 2-PG, that is 2-phosphoglycerate, and PEP, that is phosphoenol pyruvate, are similar. Enolase just rearranges to a form from which more energy can be released in hydrolysis. This enzyme requires magnesium ion for activity. One begins carbonyl groups of substrate and other involved in catalysis. The enzyme pyruvate kinase converts phosphoenol pyruvate to pyruvate in 10th step of the reaction. Here also substrate level phosphorylation generates second ATP. In the second step or second phase of the glycolysis, each molecule of glyceraldehyde 3-phosphate converted to 2 molecules of pyruvate. Much of this energy is conserved by the coupled phosphorylation of 4 molecules of ADP to ATP. The net yield is 2 molecules of ATP per molecule of glucose. The net outcome of glycolysis can be represented as glucose plus 2 NAD plus 2 ADP plus 2 inorganic phosphate is equal to pyruvate plus 2 NADH plus 2 H plus 2 ATP plus 2 molecule of water. Now discuss about the fate of pyruvate. The fate of pyruvate varies depending on the organism. In aerobic organism, pyruvate is completely oxidized to carbon dioxide, whereas in anaerobic organism, it is fermented inefficiently to an organic end product. Pyruvate is oxidatively decarboxylated to acetyl coenzyme A by pyruvate dehydrogenase complex. This reaction serves as a connecting link between glycolysis and citric acid cycle or Krebs cycle. Pyruvic acid may convert into lactic acid by the enzyme lactate dehydrogenase, and also pyruvic acid convert in the presence of enzyme pyruvic decarboxylase and alcohol dehydrogenase into ethanol. Fermentation is an anaerobic degradation of glucose, which occur in contracting skeletal muscles, facultative and obligate anaerobes, and certain tissues and cells like retina RBC. The basic purpose of fermentation is to reoxidize NADH formed during glycolysis so that it can be recycled. Microorganisms utilize a variety of organic terminal hydrogen acceptors, leading to the formation of stable fermentation products. The type of fermentation performed by an organism is named on the basis of the final product so formed. Organisms which form a single end product are said to be homofermentative, while those which form more than one product are heterofermentative. Not all microorganisms metabolize sugars by EMP pathway, that is Emmerdon-Meyerhof pathway. Alternative pathways utilized in the initial breakdown of sugars are the Ettner-Diodorov pathway, that is ED pathway, and the hexose monophosphate pathway. If we discuss about the control points in glycolysis, the enzyme hexokinase, phosphofructokinase, and pyruvate kinase directly involved in this. Other sugars may enter into the glycolytic cycle, that is galactose, mannose, and fructose directly. As for example, like galactose, which is converted into galactose 1 phosphate by the enzyme galactokinase. And this galactose 1 phosphate again converts into the glucose 1 phosphate by the enzyme galactose 1 phosphate uridynyl transferase. Glucose 1 phosphate then, in the presence of phosphoglucomutase, converts glucose 6 phosphate. Let's come to the overview of glycolytic pathway, which consists of two phases, that is preparatory phase and second is ATP yielding phase. So the first 5 reactions constitute the preparatory phase that consumes ATP, while the next 5 reactions have 2 ATP yielding steps and one reaction in which oxidation of the substrate occurs. Thank you Radio Khanchi for giving me this opportunity, and thank you to all students and listeners.